The second lecture of mitochondria, second part, we will be talking about the various names of F0, F1 particle along with the various enzymes present in the different compartments along with the different membranes of mitochondria. Okay, so F0, F1 particles are also named as Fernandez, Moran subunit. Okay, they are also named as oxysome. Okay, then they are also called ATPase or ATP synthase or synthetase. ATP synthase or ATP synthetase. So all these names are correct. In case we have been given any question in examination, you can easily say, or you can easily detect what the question tells if you have knowledge of all the names of F0, F1 particle. Okay, now F1 is the head part and F0 is the base part. Now let us talk about the enzymes that are present in mitochondria. So the enzymes, we're talking about the enzymes. So first of all, let us talk about the enzymes that are present in the outer membrane, okay, of mitochondria. For this, let us imagine that two friends, Rina and Mona, okay, they were falling from a couple tree. So it is a tree. If you don't know about this tree, you can search the internet and you will get plenty of information about this tree. Now, how can it help us to remember the enzymes that are present in the outer membrane? So first of all, R is the first enzyme. Arena is the first enzyme. So it means rotenone insensitive. Okay, NADH. Then you have the cytochrome. Okay, C reductase. Okay, now what does this word Mona say? Mona is the second enzyme which says that. So I'll write here as Rina. And the second enzyme is Mona, that is monoamine. Then you have oxidase, that is Mona. All these terms, all these enzymes will be broken down when we will be talking about enzymes. Okay, you will understand them better when you will study enzymes in the biomolecules chapter. So for now, just remember them because it's a very important and complex enzyme, and if you use these terms or you use these tricks, you can easily remember them in the right sequence or right way. They don't have any sequence, but in the right way you can memorize them. Then you have the third part, that is fall, that is fatty, okay, acid coenzyme A ligase. Okay, so you can easily see F, okay, A and L, that is fall. So fatty acid coenzyme A ligase, that means falling. And then you have the fourth enzyme, that is couple, that means kynurenine hydroxylase. Okay, and it is a very important thing to note. Next we will be talking about the enzymes that are present in the inner membrane. Okay, or the, we can say the outer chamber. So let us talk first about outer chamber. Outer chamber enzymes. Okay, so what are they? For this, just imagine that when Rina and Mona were falling from the tree, Akash and Nandita saw them and they came to their rescue. So just write it here, Akash and Nandita. Okay, now these two names will help you to memorize the enzymes that are present in the outer chamber. The first enzyme is called adenylate kinase. Okay, so A K. Now you have Nandita, which means nucleoside diphosphokinase. Okay, so nucleoside diphosphokinase, and N and D is very important in this word. Now we will be talking about the enzymes of the inner membrane. Okay, so the inner membrane enzymes can be remembered in a very easy way. Okay, for this just remember this sentence, Dipna. So suppose Dipna is a friend, so Dipna, a friend of Nandita. Okay, and she was shocked when she heard of the incident. So was shocked at the incident. Was shocked at the incident. Okay, so let us see how it can help us to remember the enzymes that are present in the inner membrane. Okay, so first of all, Dipna, the first one is Dipna, and it means that diphosphopiridine nucleotide. Okay, so you can say diphosphopiridine. So diphospho and P, have P diphosphopiridine, and then you have N, that is nucleotide. Okay, so it is very very important. Dipna. It will help you to memorize these complex enzyme names, because you have not, if you don't go through the chapter biomolecules, you cannot understand what these enzymes mean actually. So a friend, then you have the friend. This is the second trick. So friend means flavin, adenine, dinucleotide, and it is called FAD. Then you have NAD, that is nicotinamide, adenine dinucleotide, that is NAD. Actually, these are not enzymes. Okay, in the true sense, they are actually coenzymes. Hope you remember this. Their coenzymes are not enzymes. Okay, now we will be talking about this word shocked. If you look at it, you will get three things to note. First is S, that is the fourth enzyme. Then you have the fifth one. Then in the C, sorry, it should have a C, and the C is the sixth enzyme. Okay, so let us see how it is working. So first of all, 4 is S, and that means succinate dehydrogenase. Okay, then you have 5, that is the H. For H you have beta hydroxy butyrate. Then you have the dehydrogenase. Then you have 6, that is C, that is carnitine fatty acid acyl transferase. Okay, so carnitine fatty acid acyl transferase. We will be talking about the enzymes when we will talk about biomolecules. Okay, so for now just remember the tricks and try to remember them, and we'll be talking about it later in the biomolecules lectures, because now if you are watching the video for the first time, if you're starting the cell for the first time in class 11, it will be very hard for you to understand the various biomolecules and their names. Okay, then you have there is no 7th enzyme here. Okay, or one enzyme was there and we can call it acyl transferase, but it is not in the trick. Okay, so it is acyl transferase. Always remember this. Now this is the last one, that is the enzymes present in the inner chamber. Okay, so the enzymes are present in the inner chamber. The inner chamber enzymes are very important to be noted. And how can you remember this? Just remember a sentence: I am a WBCS candidate. Okay, so this is for fun only. It will help you to remember the various enzymes that are present in the inner chamber. Okay, so let us say, first of all, I means isocitrate dehydrogenase. Then you have the second one, that is A, that means aconitase. Okay, we'll be talking about this enzymes when we'll study the Krebs cycle. I have made videos on Krebs cycle in my channel. If you want to check, you can easily search it in my channel and you will get to know the various enzymes and their functions in the Krebs cycle. Then you have this WBCS, only the BCS part will be taken, and which means beta oxidation enzymes. Okay, so the B is beta oxidation enzymes. And then you have the fourth enzyme, and this is the candidate, so it is actually alpha keto. So starts with keto, alpha keto acid, so CAN, keto acid. Then you have D, that is dehydrogenase. Okay, so these are the various types of enzymes that are present in the membranes, in the outer chamber, in the inner membranes, along with the inner chamber of mitochondria. The inner chamber encloses the matrix, and you know very well that I have drawn the Christie, the finger-like projections in the inner membrane, and that is very important. In the Christie, or in the inner membrane, we have the Fernandez-Moran subunit, which is also called the F0 F1 particle. Okay, and it is also called the Fernandez-Moran subunit. It is also called oxisome, ATPase or ATP synthetase or synthase. Okay, Now we'll be talking about in details about the F0F1 particles in our um, next lecture about the F0F1 particle. And always remember that the mitochondria has its own DNA, okay, which is naked, and it has its own ribosome, but its ribosome is 55 to 70S. Okay, so you know very well what it means. The S means Weidberg's unit. Okay, now the Weidberg's unit 1S is equal to, okay, 10 to the power minus 13 semi per second per gram per time. Okay, or simply you can say semi. Okay, so that is very important, and it is called the centrifugation unit. Okay, so thank you very much for being with me till the last of the video. It was the second part, and we'll be making videos on different topics of cell, and then we'll be talking about the biomolecules again, and after that we'll be going for physiology and the plant physiology and the animal physiology also. Thank you very much for being with me till the end of the video. Have a great day.